Thank you. I don't want to. But thank you. I don't think
But you have to wear my clothes.
All right, everybody back from coffee. Okay, so our next speaker is Leili Shahriyari from the University of Massachusetts, Amherst. I'm talking about personalized cancer care through digital twin technology. Right.
Hello, everyone, and thanks, organizer, for this wonderful workshop, and thanks for the invitation, and thank you guys for joining this talk. I just want to apologize first for missing talks because I have to also go back to Amherst and teach. So sorry that I will miss your talks. Okay, so I will start with some of the background about our team, our objective, and some of our model, and then the next phase of the project, what would be. So this idea of this project started with at NCI, DOE Ideas Lab on digital twins. And it was during the pandemic in July 2022 that NCI and Department of Energy, they collaborated with each other and they hold a 5-day workshops toward building a cancer patient digital twin. So they invited many people from very different backgrounds, from clinicians to engineers to mathematicians, computer scientists, and biologists. And we were brainstorming and thinking about different ways to create digital twins for cancer patients. And at the end, 5 project teams were selected to receive seed funding, and also DOE made some contribution to 2 other teams that were selected by DOE. So our project got funded from both Department of Energy and National Cancer Institute. And the project team included me as a PI and many collaborators from many different universities. So Navid at the time was a postdoc in my group, but he's currently at Columbia University, Pamela Jackson at Mayo Clinic, Wenray Howe at Ohio State University, Ali Reza Sadpour at UMass Dartmouth, and Yang Huang Cheng at OHSU. These were the computational people in the team, and we had a set of biologists and physicians, Yanis and Adrian Lee at Pittsburgh University, Colin Sabula and Muhammad Abdurrahman at Ohio State University, and Danielle Stover at Ohio State, and Susanna at OHSU. At the time, she was at OHSU, and Eva, that was in Veteran Hospital in Tampa. So what was our goal, our objective, and what is actually our objective? Is creating a digital twin for cancer patients. As you know, that digital twin creation of it's not a very simple thing to do, and many might think that it's impossible, but you can see in the world that we are saying that how the technology grows super fast, nothing is impossible. So if we work together, collaborate with each other, that's also my call here for collaboration, we can achieve this. So the idea was that creating a data frame that included already collected so many biological and biomedical data that we have, and the EHR data that we have. And then the output of this digital twin also would be in this database. And then we have the computing part, that the computing part includes both mechanistics modeling and machine learning models. So the computing part is based on the QSP modeling. As many of you know, it's basically the compartmental models, the QSP models. So it includes mechanistic as machine learning and stochastic simulations. And I'll explain a little bit in the other slide more about the computational core of it. And then the output would be the visualization part, that basically looking at the dynamic of the tumor and how the microenvironment would change and evolve over the time. And importantly, what type of the data and when should be collected to improve the prediction of digital twin. So we know that this is the time that something would happen to the patients that might have, for example, a metastatic tumor formation, is the best time for biopsy. So it's that time. And then, so obviously the cancer evolution in the entire body, how it goes in and affects the entire body. And then we do the data collection. So try to see, to validate the model, see that if the model prediction matches with the data that has been collected from the patient. If not, then some prior info must be given by digital twin to tell us what type of the data should be collected to improve, or how we can improve the mathematical or the computational core of this digital twin. And if accepted, perfect, go and add the data to the DT. I call digital twin by abbreviation DT. If I think that I call DT, so that means digital twin. So going back to the computational core. So our objective was that in the pilot phase to develop some data-driven PDE model of the primary tumor and see that how the primary tumor would evolve over the time by modeling the cells locations. And after that, completing that data-driven of the primary tumor. So one thing that you may know that the most data that has been collected is gene expression profiles of the tumors. That means how much RNA of each of the genes are available in the tumor. I'm just trying to explain it in more simple way. And then we have the MRI images and then pathology images. MRI images mostly give us the boundary of this, and the pathology images that basically give us the cell locations. I mentioned here the pathology images because there are much more data pathology images than codex images. So what are the codex images? Codex images, they based on the new technology, they can stain the different markers and actually give you, for example, this is exactly CD4 plus T cells, CD8 plus T cells, or is it Treg cells, and what's the location of them in the tumor? I will show one of the examples of those codex images. That's, thank God that we see more of these information and these images, so we can use them to model the location of the cells. And we have really nice PDE models that we can use them for these analysis to analyze these images and create the model. So many of you here in this room using the kinetic reaction formula and basically modeling ODE or PDE between the interaction between cells and molecules in the tumor, or for any other problem, biological problem. So we are using the kinetic reaction formulas, and we develop the PDEs, and we get using these data types to basically parameterize the model and find also actually finding the model as well, also the interactions of the between cells and cytokines in the model. And then we aim to complete it and make the entire organ by basically how the tumor would interact with the organ itself. So, and then the other part would be modeling the metastatic organs. So liver and brain for breast cancer example here, that we wanted to focus on breast cancer because the biologists and physicians that we have on the team, they were more expert in breast cancer. So our goal was to modeling breast cancer. And then completing the DT with its API, the data part that I mentioned, and to be able to be connected with other projects. So as I mentioned, the QSP modeling is basically compartmental based. So each organ would have their own PDEs, and the interactions between them would be modeled by both mechanistic modeling and machine translation models. Any question here? Okay. So, so this is, it wasn't like this when I was creating, but these are basically, at the boxes here, you would have seen the name of the cells that would secrete, for example, IL-12 or IL-12. So this is the interaction between cells and molecules, the very, very simplified interactions, but more important interactions for the breast cancer. And we found this by talking with the biologists in the group and doing some literature review and stuff. So this became the sparse interaction. So importantly, mathematically, this is a large system, but for biologists, it's super small system, right? So we just say that, okay, we start with this. If it needed, we add all we can add complexity if it was required. So I don't want to go much to the equations because I know you guys are experts right here. I'm just talking about the data that are available and how we can create these equations more like a data-driven equations. How we can use the data basically, and what type of the data is out there. So this is based on the mass action law. And this is the interactions that you can create from the system of, from the network of interaction. We can create a system of ODEs. And now that I basically want to get back to the data. So what's happening here, as you all know, that we have many unknown parameters. In the biological system, our problem is many unknown parameters. These lambdas are unknown. These deltas are the degradation rate or death rates. These are unknown. Some of them we can find through literatures, but as you know that many of these death rate or degradation rate that we have is just collected for many different biological experiments. And each patient we know that is very different from another patient, right? So they actually, if these parameters might be very unique for each of these patients, but how I can use one patient and do that, because I would do overfitting if I just use so many parameters and fit them to the patients. Now, as I mentioned, we have the codex images that basically give us the cell location, but these are very small number of images. They cannot be used for parameterization of such a model with so many unknown parameters, right? Now, what we can do. So, as I mentioned, going back, gene expression profiles were the most collected data for the patients, right? There are a large set of gene expression profile data available on GDC portal, for example. If you're interested, you can look at the GDC portal, and the data has been also cleaned for individual cancer types that you can find it at USC Zena. But here, there are several methods that uses these gene expression profiles of primary tumors converted to the number of cell types. So what we are basically here, we have in the model is the players were the cells, right? And I need to know that what are these number of the cells in the tumor. How I can convert it? We can use any of these methods, like decon RNA-seq was the first method that was developed, and then CyberSort method, and then several score-based methods. After that they have been developed. Now, what we did, we created a Python package called Tumor Decon. So you can use also this package. So Tumor Decon is already connected with CBio portal and UCSC Zena. What it does, it gets the gene expression profile and some signature metrics that you need for those deconvolution method that I mentioned, and then pre-process it and you can select your digital cytometry method among those 4 methods that I showed you. And then there are post-processing and some visualization results that can be used for publications. More importantly, the Tumor Decon is, as I mentioned, connected with CBio portal and UCSC Zena. So you don't need actually to download gene expression profiles. And you can just name the cancer type over there, type the cancer type that you are interested, and then it will analyze the data and give you the percentage of different cell types in the tumor. Now, the good thing is we created also the tutorial for the tumor decon. So it's very easy if you can follow up the tutorial here and use the tumor decon Python package. And also for those of you who are interested for some of the visualization, it's basically create some visualization as well. Like for example, the percentage of different cell types in the tumor. What I'm actually showing here, the result of CyberSearch method comparison between mass cytometry analysis, that is very expensive analysis that have been done for renal cancer for 59 patients, and comparing, I think it's around 200 patients, I forgot that one, this TCGA data, the public data that's available on the GDC portals. And as you can see, the order of the cells are exactly the same. So macrophages are the top cells, and here also macrophages were the top cells, and so on. So yes, these methods, digital cytometry is not perfect, but actually we can use it when we don't have anything. We cannot just say we don't have anything and not doing anything. So we can improve this digital cytometry method, but also their performance is acceptable. So going back to breast cancer model we had, so we looked at the patient data, we clustered the tumors based on their immune profile. So we know that patients with the same immune profile usually behave similarly. And so we cluster them based on their immune profile. And for example, you have seen the cluster 1 has a high percentage of M0 macrophages, while, for example, cluster 3 has high percentage of M2 macrophages. Right, so there are very different immune compositions in tumor. Now, going back to the problem of not having data, small data, and how many parameters to estimate, what we assumed here, basically what we assumed here is that we assume that the tumors in each cluster are evolution of one tumor. Yes, I know it's really huge assumption, but we have to start from somewhere, right? I personally think it's better than just looking and estimating the parameters, for example, from another disease and apply it for the cancer. So I think we should start from somewhere and use somehow the data that we have. So that was something that came to my mind. But if you have a better suggestion, I'm super happy to hear and see that how we can basically parameterize our model, make it better, what type of the data is out there, and how we can basically overcome the challenge of this a small number of data and so many parameters, unknown parameters. So what we are doing here, we cluster the patients and we assume that the tumors in each cluster are evolution of one tumor. Now, so as I mentioned, we have our ODE model is based on mass action law, and we have the challenge of the low time data. So for the parameter estimation, I explained that basically we use the parameter for each cluster, we parameterize separately. And then what we do, sensitivity analysis, what else we can do? So we perform sensitivity analysis to find out what are the most sensitive parameters, and then focus more on those sensitive parameters to find the values. So we looked at our ODE model and we did the sensitivity analysis, and you can see the result for each cluster in each row. And we found that most sensitive parameters, no surprise, related to the cancer proliferation of the cancer cells or death rate of the cancer cells. In general, the death rate of the population, or by the death because of the cytotoxic T cells, or also the proliferation of cancer cells because of adipocyte or IL-6, or the general proliferation of cancer cells. But setting aside the most sensitive parameters that are related to cancer and going back that what are the other most sensitive parameters, we found that interestingly, adipocyte comes up. The death rate of the adipocyte and the proliferation of adipocyte become one of the among most sensitive parameters. And then after that, again, the sensitive parameters for the total cells, we can see that the proliferation of cancer cells by adipocyte, proliferation of cancer cells by IL-6 become the most sensitive parameters for all clusters. And then we varied these sensitive parameters and to get the confidence interval and also see that the effect of them. So we looked at the dynamics by varying these sensitive parameters. And we saw that for some cases, we see a dramatic change. For example, if we vary the parameters related to a death rate of the cancer cells by IF and gamma, but interestingly for one of the clusters, not all clusters. That shows that different immune compositions shows the different behavior of the tumor. So it's very important that finding a treatment for a specific patient rather than other. As you can see that some of the treatment work for some patients, but not for other patients. And this clearly showed that. The only change we did for one of the clusters that they have the different immune composition, we just changed the parameters that was related to death rate of the cancer cells by IF and gamma. So that one, we didn't have a time course data. So we looked at the mice model that we had the time course data for it. And so we looked at the digital cytometry again, using digital cytometry to get the different percentage of the each of the cell types in the mice. And we collaborated with Alireza Azadpour at UMass Dartmouth. They did the optimization part using topology optimization to find out what are different values of the parameters. And then again, we did the sensitivity analysis. This is mice data. As you can see, setting aside the parameters related to cancer, what we have seen here is more IL-6 parameter came out. And then for one of the mouse becomes the parameters related to the adipocyte, and IL-6 is coming up here again. So the parameters that related to the proliferation rate, secretion rates of the IL-6 by adipocyte is coming up for these mice models. So as you can see here, suggests that adipocyte and IL-6 are something that we can target for the mice. So we worked with Susanna, as I mentioned. She was at the OHSU at the time. And this is the codex images that are so valuable to have, basically staining the location of different cell types in the tumor. So we had we had the tumors that were in the large, small sizes, so early tumors, and then the large tumors here, that basically the later. So and then look at here, these are different cell types. As you can see that immune cells were depleted over the time, while cancer increased a lot, and then adipocyte decreased a lot. So almost all cells decreased to the point that they vanished, but the cancer cells took over the entire environment. So the image. So here, for example, the location of cytotoxic T cells.
But the gray is what
the gray is the tumor microenvironment.
Okay.
The entire tumor.
It's hard to see.
So the blue points. So each of them has blue points. Each blue points, for example, here is Treg cell. Here is cytotoxic.
Yeah, no, I couldn't see the blue one.
Yeah. Okay. Sorry. This one is T helper cell.
Yeah,
T helper cell. Yeah. So there are very small number, but not many. But there are something. So going back to the problems that we have and we need to solve together, and also looking for collaborator, here is that you can see the boundary of the tumor. It's so non-smooth, right? So if we even using MRI images for breast tumors, and we want to use it to our model, is that non-smooth boundaries. We couldn't solve this problem. I mean, for simplicity, we are just using the oval, the largest oval that we could fit in the boundary and have all the cell types in it. So I'm super interested if you guys know that how we can use both kinetic and mechanical interactions together and have the boundary that's not as smooth. Another open problem that we are facing is that right now, there are two small tumors, because tumors are not basically really one location that it grows in the organ. So what happens is that we have a small chunk of the tumors in the small area, right? And they evolve and they merge together, right? So I'm also we are not able to basically model this merge by the chemical interactions that we have. So we have biomechanical and chemical interactions. So we want to basically be able to merge these tumors numerically and solve it, right? So I'm very interested if you have ideas on how we can overcome this challenge.
So this is section.
Yes, this is the 2D sections. So this is one slide of the tumor that they analyze. Yes. So the images were analyzed by Yang Huan Chang at OHSU, our collaborator in bioengineering department. So they looked at the different thresholds because these are basically the biomarker, and each of the cells over there, each of the grid actually says that how much of these biomarker have been expressed. And you should know that, for example, what are the biomarkers for Treg and find a good threshold for it to give it that. So the image was analyzed by Yang Huan Chang, and he gave us the grid of the what points we should use in the model. So we changed our ODEs to PDE and we used Robin boundary condition. And then we looked at the results. So we used early tumor results as the initial condition. And we used the ODE that was parameterized by another experiment that was only gene expression data. It was not this experiment, but it was the same mice model. So interestingly, the PDE showed the exact depletion of the most immune cells that we had. As you saw in the data also, the immune cells were depleted. And then interestingly, adipocyte is also depleted, but it's just in very small steady state conditions coming to that. And if you have seen that adipocyte were coming down, but not depleted in that time frame. And we looked at the most sensitive parameters, and diffusion rate of the cancer cells, no surprisingly, became the most sensitive parameters. Oh. And then the other sensitive parameters were influx of the cytotoxic T cells, basically causing the cancer to degrade, and then cytotoxic T cells, the influx source of it, and then obviously the death rate and proliferation of cancer cells. So any questions? Now I want to shift a little back on colon cancer. Why is it just colon cancer? Because a study, there was when we were just modeling, we found very interesting data for colon cancer of patients, the codex images. So these are again codex images, but this is not mice model. This is the patient data. As I'm saying that these, the good thing is that the number of such data is increasing, but it's not in the level that we can use for parameterization of the model. So this is the location of each of these. So we looked at the data, we found the location of different cell types, discretizing the location of these cell types for colon cancer. So, this is the movie I want to show you. This is the one that so the model was parameterized using the TCGA data obtained from GDC portal for colon cancer. And then the data that I showed you here, the special location that have been used for the initial condition, the location of the cells in the tumor. And if the movie, okay. Sorry, it's supposed to be a movie. Actually, it doesn't show that's movie. Okay, so assume this is the movie. So what you will see is that co-localization of macrophages and cancer cells together. It seems that they feed each other. They give each other feedback. And they basically, you will see that each location cancer cell goes, macrophages goes there. And also they basically feed each other. And as I mentioned, we didn't fit this codex images that were available for the patient. But the result of the paper also showed that co-localization of cancer cells and macrophages. So the sensitive parameters, we looked at the most sensitive parameters. And now I want to give you some background here. So obviously, in the sensitivity analysis, we always looked at what are the sensitive parameters for cancer cells, which one of them basically make cancer cell increase or decrease. But here we look more, actually. We looked at the, not only the cancer cell parameters, but also the ratio of cancer to immune. This is becoming much more important in the context of the immunotherapy. You want to see that how many immune cells are actually inside in the tumor microenvironment. And then also looked at the velocity, the most parameters for changing the speed of the cells in the tumor microenvironment. And then the angle of the movement. What are the most sensitive parameters that's causing the different angle of the movement? And those that you see in the red box are the intersection of these parameters. And you can see the intersection of these parameters. The first parameter that's coming a lot is macrophages, diffusion rate of the macrophages rather than the cancer cells. So again, the interesting part is that macrophages seems to have, and it's not only us as a mathematical model, we're saying macrophages are very important. In many, many publications in colon cancer also shows that the role of macrophages and how important is it in the tumor, in the response to the treatment and tumor's progression. And after that, we see the diffusion of dendritic cells. That was interesting and unique to the colon cancer. And we found several studies that basically talk about the role of dendritic cells in colon cancer. Now, so this became our workflow so far in our lab. What we do, we looked at the tumor microenvironment, looking at the location of different cell types and the number of them. And based on that, we cluster the tumors because this is the data that we can find and look at the cell interactions and molecules using the kinetic reaction formulas to model the interactions between them. And then analyze this model without any treatment, see how the tumors grow, how many clusters we have, and how each of these cluster behavior. And then after that, using the data of the treatments, adding the treatments. So we parameterize the model without treatment and then add the treatment and then see how each cluster would respond to the treatment. So we don't fit to the data that's coming for the treatment, right? So our goal is that minimize fitting any sort of fitting as much as we can so we can be able to basically predict the response to the treatment. Now, going back to the colon cancer, we went back to the ODE model because we didn't have that much of the parameter for the PDE to start with. So we started with the ODE model. And then, as I mentioned, we did parameterize the model using the data that was not for treatment. For the entire, the first biopsy of the patients that they had, they removed the tumor. This is the GDC portal TCGA data. We found the parameters of the model. We looked at the progression of the tumor. And then after that, we added the treatments for each of the clusters. So this is the interactions between the treatment and tumor microenvironment, the bold part that you can see that we added later on after the parameterization of the model. So interestingly, we looked at the fall-free treatment, which is the most common treatment for colon cancer, is a chemotherapy treatment that includes 3 drugs. And then 5-FU is one of the main drugs in the treatment, and there are 2 other drugs in the treatment, Novaricon and Citron. I forgot the name. Okay. Sorry. I don't. So yes. So it was 5-FU and LV, Novaricon, and irinotecan. I don't know. So however you pronounce it. And then we looked at these 3 treatments, their interactions, and we saw that in 5-FU, you can see that the tumor would decrease over the time, the tumor cells and the total number of cells, but it goes back again. And then you can see if you have all fall-free treatment, the common dosage of them is for some patients, for one of the clusters, we see that it comes down in the terms that really down But for other clusters, no, it's just a little down and then comes back up. So it shows that for one of the clusters, that was basically cluster 3, that seems that this treatment has a little good progress versus the other clusters. And what so we looked at the start of the treatment time. After 1 year or separately after 5 years, how it basically works in these treatments. I'm going to give here again a background a little bit. What we see and hear in the news mostly is that if you have early treatment, it's much better for you than wait until the last. And then usually some of these are chemotherapy and surgery and so on. So the most common treatments that we have are chemotherapy, surgery, and radiotherapy. Now, I personally had this question, how do you know? How do you know my tumor, how long would it take to come to the stage 4, right? I know from a stage 1, I have higher survival, right? They say that, okay, you have 10 or 20 years survival rate, versus then, for example, a stage 4 cancer has whatever, 5-year survival rate, right? Now, how do you know that my tumor in 10 or 20 years gets to the stage 4, or versus, for example, 5 years gets to the stage 4? You don't have that data for patients, right? You don't know that how long would it take for this specific patient that the tumors have been developed in. I mean, when was the initiation of the tumor for this patient? How do you know that? I personally don't know. So was it, I mean, did it come to your mind also? I mean, that was something that I was struggling. That when they tell me come and start the treatment early, how do you know that this treatment actually lower my expectation of life or not, or increase it, right? So that was my main question. And then, so I said, okay, let's start. We know that this is the different compositions of the immune cells in each of these clusters. Let's start the treatment 1 year early versus that, for example, simulating that for 5 years later on in the life. And actually, so what happens, and the conclusion, just because the drawing is not really good to explain through the drawing, what I have seen for the sum of the clusters is actually true. So it's better to have the early treatment to have smaller number of cancer cells. But some of the clusters are not actually validated. Some clusters become higher concentration of the cancer cells, less as immune cells, and then the growth rate of them are much higher than the other clusters. So we should have some sort of knowing that what type of the immune composition the patient has and what type of the treatment that we want to use to say that definitely that, okay, early treatment is much better than later on treatment. This is just the model says that, right? So I don't know, actually, this is still my puzzle that if I have the early stage tumor, would it be better to have starting the treatment now or waiting? And as we know that, for example, Dominic, you're modeling also cell competitions, right? So the problem is that cancer cells, when die, and in the model of a stochastics model that we have, we assume that the cancer cells have higher fitness than the normal cells, right? So most of these treatment, what they do basically, they create some sort of wound inside the tumor microenvironment. They're causing the cells to die, right? Now, when the cells die in the tumor microenvironment, then immune cells become active, right? So immune cells start sending the proliferation signals and tell the cells to come and replace themselves. Now, if the cancer cells have higher fitness, right? So they have much more power to overcome the normal cells. So they divide with much higher rate. And then what happens after the treatment, the higher density of cancer cells stays there. And now this new tumor that reoccurred, it has much higher concentration of cancer cells than the original one. And it's basically grow much faster. I mean, if you do a simulation, both stochastically and also the ODE model, by this assumption that assuming the cancer cells have higher fitness than normal cells. Okay. Now, what we did with this data, as I mentioned, we had this treatment for free treatment. And we looked at if the tumor would be visible at the time that the physicians look at the scan of the person. So we looked at the one threshold that it was the smallest tumor in the data, and it says that if the size of the tumor was more than the smallest tumor in the data, then we can say that it is visible. So with this type, it's just say that the tumor reoccurred or not reoccurred. So with this classification. And we saw that with the first time of the follow-up, we had a good performance, but not perfect performance. But for the second time of the follow-up was the worst. And the reason was that basically we didn't model the reoccurred tumor because we didn't have the gene expression data of the reoccurred tumor. We had from original tumor. And then the patient might have the reoccurred tumor, and after that, some follow-up or other treatment, and we didn't actually model the second treatment because we didn't have the data of gene expression for a while for that. But also the first one that we had, it was okay, but not very good. So it needs to be improved. How am I doing with the time?
Another 5 minutes maybe.
Okay. So as I mentioned, I wanted to say we are looking at the MRI images and getting the boundary of the tumor, and then the open problem that I already mentioned, that we have the non-smooth boundary and also the merge of different tumors, small tumors with each other. And then I'm collaborating with Kate. Recently, we started some collaboration with Kate at Kentucky University to get data for colon cancers, looking at the velocity and the movement diffusion rate of the macrophages and other immune cells in the because we have seen that macrophages diffusion rate of them are most sensitive parameters. So right now we are focusing on most sensitive parameters and collaborating with experimentalists to get these data for colon cancer from Kate and from Yanis for the breast cancer. So I just, with the small time that I have, I just want to mention about another project that I'm working with people at, so this is also, it's part of the Digital Twins Ideas Lab that I joined with the NCI and DOE. Basically, that one started with, I also was a part of another group, Chi Wang at University of South Carolina, Tanvir at IBM, and Jundeng at Yale University, and Merke at Oslo, and Pamela Jackson at Mayo Clinic, and Eva at VA. So this model is machine learning model, completely machine learning, and looking at the patients, how they are close to each other, looking at the graph of the patients and looking at if the patient is close to this one. So basically saying that, okay, the patient, the closest neighbor basically give you the same similar behavior that the other patients that we have in the data. As you may know that IBM started the project way, way earlier that basically trying to using EHR data to predict the basically the response for any sort of treatments, not only and any sort of diseases, not only the cancer. So they have worked on this problem many. So the main core of this coming from IBM and Tanvir, the idea of looking at this patient. But I personally think that we need to merge together. So looking at both machine learning and mechanistics modeling and merge them together and basically find a solution. So these two projects can be merged together in future. So some of the challenges, I think I already mentioned all these stuff. So the lack of the data, but another challenge that I want to make it also highlighted here is how to model all the treatments and all drugs. Because patients not only have this specific chemotherapy or radiotherapy that they have also different treatments and drugs. Even those painkillers have a lot of influence on the tumors. So they suppress immune cells. And those NSAIDs, and also you can say that, okay, these are just painkiller, but they have effect. And the other problem is also assuming we have all data and we have a perfect model, there are also problems that we don't know what drugs the patient get. They might not give it to the EHR, so it's not available there in the data. So these are the, and also one of the important things, how we can add those treatment and their pathways to our QSP model. This is one of the main challenges as well. So by that, I would like to thank NIH and Department of Energy for funding, and my team members. I presented all this, but they did the work. So thank you.
Thank you. Questions?